Hi, girls. This is adaptive immunity. So it's the second part of the immune system. Um, a quick comment, if you want to review the first part, these videos are pretty good review of the inflammatory response, which is the hardest aspect of the first part of the immune system. First part of the immune system is called innate immunity, and the part we're going to do now is adaptive immunity because it adapts and becomes specific to specific types of pathogens, specific viruses and bacteria. Okay, quick vocabulary. Every bad guy, be it a bacteria, a virus, also can be a fungi, it can be a parasite, they're all called pathogens. Pathogens are the names of the invaders, the bad guys. On the surface of their cells, they have proteins, just like our cells do, but their surface proteins have a special name, antigens. So all the bad guys are pathogens. On their surface, they have antigens. The antigens are important because it's the way that our cells identify them as foreign. A surface protein is kind of like a label. Okay, now the famous, often talked about antibodies are something we're going to look at. So an antibody is overall simply a protein that our bodies make which has a Y shape. This is one antibody. This is another antibody. It's a Y-shaped protein. I'll get a more realistic picture in a moment, but the reason why this one is so schematic is because it can be useful. The antibody has two tips. Each tip of the Y is an antibody tip which has a special binding site or a special receptor. It binds to an antigen, a bad guy. So let me just go back really quickly. This picture is incomplete. It shows us just an antigen as a loose piece. Here the antigen is the red part, not the orange, but the red. Here the antigen is the darker purple part. You have to remember that the antigens are on the surface of an entire bacteria or an entire virus. So if this picture were more complete, there'd be a whole virus attached here. This would just be its surface protein, right? A whole virus attached there. Same thing here. There'd be a whole bacteria or virus attached to this thing. Here they're showing us just the antigen because it makes the diagram much more comprehensible and easier. So to be honest, there's a little bit of ambiguity between the term antigen. Sometimes it means just a surface protein. Sometimes they use it to refer to the entire pathogen. So don't worry about that too much. What's really important here is two things. Each antibody has a little spot, a little tip, which is a binding site, which is ability to bind to an antigen, right? Here it's about to bind to it, here it's bound to an antigen. So these binding sites can attach to antigens, right? And therefore attach to pathogens, attaching to the surface proteins of pathogens. Same thing here. This orange part is a binding site. Here, this binding site is just linking up to an antigen. Here it's just attached to it. Equally well, it can be called an antigen receptor. And so what's important is that there are specific antigen receptors or specific antigen binding sites which bind to specific bad guys, specific antigens / pathogens. So notice these different colors are not meant just to be pretty, but to show, and actually the different shapes also, to show that there's a specificity. So for example, maybe this is, I don't know what illness, the common cold. And so this would be an antibody for a common cold virus. Maybe this antibody here is an antibody for, I don't know, some other illness, some other bacteria that attacks us and infects us, like strep throat, let's say. And so this is the strep throat antigen, and this is the binding site force that binds to strep throat. And so this antibody is a strep throat antibody, okay? So there are different kinds that are specific. Just to look at the specificity really carefully, these would represent all types of antigens, all types of surface proteins of bad guys, right? Different illnesses, different bacteria, different viruses. And look, this antibody is very specific. Is it going to fit with this? No. Will it fit with that? No. It's got this shape which will fit with just one antigen, okay? The shape is really representative of a chemical complex that the two can form, but the shape is a nice image of how they lock together. This is just another image of the same thing. These are all antibodies. See their two tips, right? Each one has two tips, and the tips are specific for what type of antigen they bind to. Okay. this is the same exact thing. You can just look at it. I just want this to be very vivid for you. That the two tips of the antibody make it specific to bind to only one type of antigen / pathogen. Okay, so this is a summary. Everything in red is a bad guy. Everything in blue is a good guy. And notice that these are the bad guys, and they're binding to the good guys. So take a look at this diagram and now try to label everything on your own. Make sure you understand it. Okay, here's another picture of the same thing. It's showing a bacteria that has several different antigens and different antibodies binding to it. Don't worry about this too much. This is just an image of what an antibody would really look like. Remember that proteins are huge macromolecules. So instead of looking like those perfect little schematic Ys we've been looking at, they would look complex, something like this. That's just to keep in mind. It's a massive macromolecule. Okay, you should stop and read this. Let me do the first sentence with you. How does a cell identify another cell or another substance? Do you remember? This was from the very beginning of the school year. I hope you remember. Surface proteins. Our glycoproteins, which are on the surface of our cell membrane, are like our ID. Okay, you can read the rest on your own. And same thing here. Read the rest on your own. This is just some general background. Okay, so now let's look at white blood cells. This represents 5 types of white blood cells, and 6, 5, 6. These are kind of 2 subcategories. The ones we really need to know are, first of all, this one. This is the one we've been studying all along, the macrophage. This is, in a silly way, I like to call it a Pac-Man. It doesn't have the shape of a Pac-Man, but it kind of does what a Pac-Man does. It ingests, right? It engulfs and then digests pathogens. So that's why Pac-Man is a very good image for it. The engulfing and digesting of pathogens is called phagocytosis. Right. A neutrophil is a new one. We haven't learned about it yet. It's also a phagocyte. I call it a little Pac-Man. So it's a phagocyte that's a little Pac-Man. Don't worry about this. We're not really studying this. I'll tell you what it is real quick. These little granules contain different kind of chemicals which are toxic to pathogens, and they get released. But you don't need to know that. So you do need to know that a macrophage is a big phagocyte, and neutrophil is a little phagocyte or a little Pac-Man. And we're skipping these, and we're beginning to study lymphocytes. Lymphocytes are called B lymphocytes or B cells, and T lymphocytes or T cells. There's 2 types, and these are the really quote-unquote smart white blood cells, or the white blood cells that can target a specific type of pathogen and attack that pathogen in particular. And so they're what form antibodies and therefore immunity. So now we're going to take a look at the B cell or the B lymphocyte in particular. And you'll recognize hopefully what these are. These are our antibodies that we just learned about. And now you find out where they come from. This is a B cell. This is the white blood cell called a B lymphocyte. And it has antibodies all over its surface. It's got surface protein. It's got antibodies for surface proteins, right? And remember, it's not just a circle, but it's a sphere. So it has many antibodies. And remember that antibodies are specific. So in this picture, they made different images, right? The different shapes to represent different types of antibodies. And now you know that different B lymphocytes, each one has a specific type of antibody, and therefore a specific type of antigen it can bind to. Okay? So this is, again, an image of 3 B cells, and each one is coated with surface proteins that are antibodies. And what happens is that here, this represents the antigen, the bad guy, the pathogen that has entered your body. And the B lymphocyte has found its matching pair. It has found the antigen that it can bind to. And therefore, this B lymphocyte has just been activated. It's just like, oh my goodness, my enemy is here. I have to get to work. So it's been activated and it's ready to start fighting. This one has no antigen or pathogen attached to it. It hasn't found any bad guy that it matches up to. So it's still more or less dormant. It's just sitting in a lymph node there as a patrol, waiting to see if anything will match up to it. If nothing matches up to it, it remains dormant. If something matches up to it, it'll get activated. What's happening with this one? This is also a B lymphocyte with yet another type of antibody on its surface, and this is made to represent the antigens that can bind to it. And so obviously this B lymphocyte is also activated. Okay, this is a little bit more of a detailed picture, so you can, actually I made it bigger here, you can stop to look at it, right? It's just a reminder that antigens are where? They're on the surface of pathogens, of bad guys. Please don't worry about this epitope. I should have crossed that out. Don't worry about it at all. And B cells have antibodies on their surface. And what is it that bind together? The antigens, which are part of the bad guy, bind to the antibodies, which are part of our B lymphocytes, our B cells. Okay, this we already did. You can answer that question, I'm sure. And here we go. We're going to go one step further. In this picture, there's 3 types of B cells, and each one, the color is made to represent their specificity, a different type of antibody that they each have. This one has the gray antibody, this one has the royal blue antibody, and the turquoise antibody. The black dots are made to represent antigens, bad guys. Pathogens / antigens have entered the body, and look what happens. This antigen has paired up with this antibody. So this is the B cell, which has the code and has the correct kind of antibody that can bind to this particular bad guy that just entered the body. These are no help. They don't have the specific code for binding with this antigen. This one also, it just can't bind to that antigen. They don't fit together. This is the big help. This one with the royal blue antibodies somehow binds to the antigen that has just entered your body. And so what does it do? It becomes activated. All right, so that previous picture was a zooming in on this. Here we have the same thing going on, and you see this is the one that binds to the antigen. This one becomes activated. What does it do when it gets activated? It clones itself. It divides itself over and over and over and over and over. So it divides itself in 2, and then divides itself many times. So there ends up being many of these cloned B cells. So there's 2 reasons why the clones don't look like their parent cells, even though they're genetically identical. They are the identical cell. The first reason is that they have secreted their antibodies. So their antibodies are all loose in your blood and in your body fluids. And so that makes them look different, because these are kind of pretty with, they're wearing their antibodies like little antlers, and these have lost them. That's the first reason. Second reason, you notice these are bigger. And what they have here is endoplasmic reticulum. Hopefully you remember from the beginning of the year when we reviewed organelles that endoplasmic reticulum is the organelle needed for making proteins. And guess what antibodies are? Proteins. And so these all have the job of producing tons upon tons upon tons of antibodies and secreting them and sending them all over your body through the blood and then through the interstitial fluids. So I gave them the name of little antibody factories. And that also explains why they look different, right? They're secreting their antibodies. They have extra, extra ER, rough endoplasmic reticulum. Okay. So that explains this very same thing. Now I want to explain to you what antibodies do. So when many antibodies are released into your blood and your fluids, they bind with both of their sites. Remember, both tips of the Y have a binding site or an antigen receptor. And when they do so, look what happens. They end up clumping together, precisely because they have 2 binding sites. This antibody has bound those 2. This antibody has bound those 2, and so on. So they end up clumping up. And the result will be a massive clumping up of antigens / pathogens. And why is that helpful? When they've been clumped up, they're not able to invade your tissues and attack you successfully, number 1. Number 2, it's very easy for the macrophages, the Pac-Man, to find them and devour them. And so here's just another picture of the same thing. So, the clumping up has a fancy name. It's called agglutination, like glue, right? They clump up together, and that way they're kind of like all rounded up in one area, so they can't invade your tissues and attack you. They're now harmless, and the macrophages will easily gobble them up. Or they can get, an individual one can get coated with antibodies, and therefore it can't enter your cell. It doesn't have the capacity to enter into your cell anymore, so it's called neutralized. It's kind of like it's been handcuffed. It becomes